So with that, let's move along in the agenda. And now we have Dave Tuveson from Cold Spring Harbor Labs, who will talk about organoids. And this came from a meeting that JR, Chuck, and I had with Dave up at headquarters at Cold Spring Harbor Labs, where he was very excited about a new technique. So David will give us a quick overview of that.
Thank you, Lee. Good evening. Cold Spring Harbor and Jackson Labs are excited to be science partners to SWOG and all of you. Our job is to bring new ideas to you that can be considered in the way that we study the cancer problem so that we can come up with better therapeutic and diagnostic approaches. And this new approach that Lee heard me get excited about is one that we are particularly so right now. So on the first slide, you see a picture of Hans Clevers, who's the head of the Dutch Science Foundation right now. He was one of the recipients of the Internet Biology Prize last fall. So what is a cancer model? A piece of tissue on a slide that we've heard about for the past hour is not a cancer model. That's evidence of cancer. A cancer model is something that's alive, that you can perturb, that you can learn something about your perturbations so that you can adapt your information. And so the models that we've had over the past have been the HeLa cells, the nude mice with xenografts, the genetically engineered mice, things of that nature. We're trying to model, obviously, what our patients have. And we're looking for representations of the curiosities and the mysteries of human cancers, the multitude of molecular alterations, the heterogeneity therein, both at the DNA and epigenomic level, and of course the tumor microenvironment, which is very poorly modeled. We want to know what drugs work and what drugs won't. We heard a little bit about toxicity, and that's a huge problem too. And so again, the third issue, though, is we want models that are feasible to use in the nice randomized trials that we're charged with running through a cooperative group like this. On the bottom of the slide, though, are the things that we would love to have in our models. We would like cancer models to be prepared longitudinally from our patients so they can represent the evolution of disease, the before we treat them, while we treat them, and when they relapse, as we heard from some of the talks today. We would love from our models to tell us the therapeutic index, meaning what kills the cancer cell and leaves the normal cell behind of that tissue, and then of the marrow compartment, the GI tract, liver, etc. And of course, we would like models that would tell us about the mystery of the tumor microenvironment. That's been hard. So I'm just going to tell you about a new tissue culture model which you can prepare from primary tissue. It's been published by several groups, Hans Clevers chiefly. There have been variations on the theme by several groups from the States and Asia too. And so on the picture is a poor representation of the pancreas with a loop of bowel and a gallbladder here. The red star is the pancreas tumor, and so you can prepare tissue fragments from the tumor itself or from the normal tissue. And in this case, we're talking about the duct, the normal duct and the tumor. These two gals shown here, their photograph are the ones who actually got this off the ground, Sylvia Fernandez-Boge from Hans's lab and Mona Spector from mine. And from both of these tissue fragments, we prepare what we call organoids. We call them organoids because when we transplant them back into the tissue from they came, they turn back into that tissue. We don't have to add Yamanaka factors. We don't have to add telomerase or any of the things that you've heard about and maybe you use in your own lab. So here's the normal pancreas from a mouse. Here's pannin, the pre-invasive malignancies or pre-malignant malignancies that you would see in humans or mice. And here's a proper tumor. And so you can prepare from this tissue within 48 hours something that looks like this. It looks like a balloon, and it's hollow in the middle. It essentially floats and lives in a gelatin substance you can purchase commercially called Matrigel. You can make it yourself. There's a lot of variations on the theme. And they all look the same. So under the microscope, you can't tell them apart. You only can tell them apart when you transplant them back in. And when you do that, when you transplant normal tissue, it makes normal ducts. Pannin organoids makes pannin. Tumor turns into a high-grade pannin that does invade and metastasize. And when you look for markers of the tissues, you find them. And so the pannins by chemical stains have mucin accumulation. You can stain them with Alcian blue. You can use antibodies against known biomarkers of pannin evolution, such as this mucin here. With these organoids, you can now do deep science. So you just heard about the limits of detection with today's best technologies, which keep getting better. And the technologies keep improving and getting cheaper to be more feasible. But the weaknesses or the limitations that you heard about from Chris and you heard about from James and others are multiple. And one is, if you have contamination of your sample by normal cells, that makes it hard. If you have multiple clones, it makes it hard. Well, you can make an organoid from one cell. And so you can actually study one cell at a time, and you can pick 20 out and actually analyze all 20, one at a time, because it grows as a clonal population when you start it. So you can do real biochemistry, so pathway activation, look at RAS GTP loading, look at a transcription factor that we fancy called NRF2, which runs the antioxidant response in multiple cancer types, including pancreas. You can do gene expression profiling. Some of the NRF2 targets are shown here. And you can study biology or pathobiology of cancer. And so some known transcription factors relevant in pancreas and biliary development are shown here. And on the right of the slide are some markers that go up when a normal ductal cell turns into a pannin cell and then a cancer cell. And they go up orders of magnitude. And you can see that in a cell preparation that grows in matrigel. If you try to do this in 2-dimension, it will never work. The cells cannot survive that, particularly normal cells. So what can you do with this? You can now study the tumor microenvironment that I talked about. So you can grow the organoids by themselves or admix them with the fibroblasts and the other normal cells that populate every tissue type in our bodies. And in that case, you can get the stromal fibroblasts, which are called stellate cells in the pancreas, to lay down this cement, the collagen, the hyaluronic acid, all of the ingredients that are part of the extracellular matrix, as shown here. And you can stain them with the same chemicals we do in the pathology lab, H&E, Mason's trichrome. You can look for evidence of the tumor microenvironment after you transplant it, and you'll see the tumor microenvironment coming in. And on the right of the slide, you see this multicolored picture. This is actually the stellate cells communicating with the organoids. You can pull out all the ingredients to feed them, and they live off each other. They're a real lichen, which is probably a big component to tumors that is, pretty much impossible to study outside the body. And so I'm a medical oncologist like most of you, and I only went into this because I was looking for better drugs too. And so what have we learned so far using this system? We haven't done high throughput anything, but we have done high content few things. And this is one of them. We've just looked at known mediators of the RAS signaling cascade, are the organoids differentially sensitive or resistant to drugs that we could get our hands on today? And essentially, as you shift to the right, you're going from normal to malignant. And so the more malignant your organoid is to start with, the more resistant you are to the same pathway inhibitors that you just heard about on the last talk in melanoma trials. AKT inhibitors, MEK inhibitors, et cetera. And Mariano is the one leading this part of it, and he's here to actually learn from you, and he'll go to the GI meeting in an hour. And so he's multicolored them, red and green, and so the normal in this case is red, and the tumor one is green. And he's got a drug that actually makes the tumor ones go away better than the normal. So now you're talking about therapeutic index. What about in the humans? We're not here to study mice, we're here to study our patients. You can do this with humans too. It's a bit more involved, but essentially you're dealing with the patients who get resected because they have a localized tumor of their pancreas. And working with Hans's group, you can make them from normal ducts, you can make them from tumors, and some examples are shown here. And so the questions that we have with this technology, because we now have a viable living tissue culture system that can be sampled once, grown up, frozen down, and then thawed out to grow again, we're asking, is that a sample system that could be very useful for some of the questions you have here in your trials? But also, is this a system that can now capture, as a living entity, things like circulating tumor cells? The early evidence from other types of cancers, particularly prostate cancer, is yes, you can grow CTCs using this technology. Grow them up, freeze them down, and then use them when you're ready to. One of the really big questions we have that we're hoping to ask with Edison's group at Jackson Labs is if you were to compare organoids to patient-derived xenografts, do they give you the same information? Tumor heterogeneity, therapeutic responses, feasibility, tractability, the ability of many people to use them. So that's a critical question that we need to address right now, and with the support of you all and the Hope Foundation, we're going to do so. So we just heard about the limitations of what we can do. Can we see the mutation? This is one way to do it. Can we see the heterogeneity? We're going to address that. What about toxicology in normal cells? We can now grow normal cells. You can grow normal liver, normal marrow, normal whatever you want. And then finally, you're going to hear the plenary talk after me from Mike, who's going to talk about some of the amazing things the immune system is now doing. Well, what's the antigen? How are you ever going to figure that out? Well, what if you could expand the population of cells so that you had 1 million cells, target cells, or 10 million, so you could actually find the peptides that might be a better way to run your T-cell dependent immune therapy trials. So this is the new approach. I'm hoping that in a year or so, we'll stand up here with clinical data, patients that we've prepared organoids from, things we've learned from them. But it's certainly the most new exciting thing that we're doing. Thanks.